get done need to have good controls. And controls to scientists are one of the standards that we need to be able to measure and compare things to. Our controls can be against our conventional crops as well as other things that are missing items that are being tested. However, nutritional and feeding studies are particularly difficult because the nutritional community knows that they cannot increase the amount of an individual food without depleting other nutritional requirements. So actually being able to draw conclusions from certain feeding studies are really, really challenging because we don't necessarily know if it's the increase of the item that we think is really that is being tested, or whether it's a decrease in all those other important nutrients that are needed to create a healthy organism. This is one of the biggest challenges that the whole food industry, not just the GMO industry, is confronted with, that nutritional feeding studies are hard. So how do we go about this? We have to think about what we already know and what we accept and be able to try and design things based upon certain ingredients. But whole food studies are really difficult.
Yeah, I would just quickly like to respond to that because the design of the studies are not appropriate. I would point out for anyone, if you want to look at the Seralini study, just look at table 1. Because 8 years before in that same journal, Monsanto had published, and their methodology of what Seralini did is they basically reproduced it. Rather than run it for 90 days, they ran it for 2 years. And they were actually more accurate because when they looked at the control diets, it's the same amount of corn, but supposedly non-engineered ones. Well, the control diets, you have to test to make sure that there's no GE in them or to make sure that there's no residues of glyphosate. Seralini did that. Nobody else did. So, for example, in the same journal, Food Chemical Toxicology, in late 2014, a study was published from Canada. Roundup Ready canola and then non-engineered canola, with conventional diets, they found no difference. Well, Seralini's team went in, they bought the same control diet, the same batch, right? And they tested that batch of control food. And guess what they found? 200 parts per million of glyphosate, 100 parts per million of AMPA, which is a breakdown product of glyphosate. 27% of the corn in there had Cry1AB, and another 25% had Roundup Ready. So in the control diet, there was not only GMOs, but there was glyphosate residues. So you do need to actually test. They should be accurate. In terms of the protocols, that's exactly what this global 8-year process was. There were actually expert consultations on how to do safety assessments for engineered animals. I was actually an international expert at that. We came up with a long set of protocols. I could go through them for allergenicity and other things. Again, those were published globally. The U.S. cannot meet those standards. And finally, I would say for adequate controls, we do need independent research, but because these crops are patented, the companies, the scientists have to get approval from the companies before they can do the studies. If the company doesn't like your study, you can't do it. So there's no true independent research on any of these crops. Even here at Cornell, they have to sign an agreement with Monsanto, and none of those agreements have been made public.
For the interest of those in the room who do not know the results of the Seralini study, which we have been discussing at great length, could I say correctly that it showed an increase in liver and kidney damage and an increase in tumors? Is that correct?
Yes.
Okay. Now we go to audience questions for the supporters. Do you think it is necessary to understand how the inserted gene interacts within the entire genome? Who wants to take that?
That's for the supporters. But
for the supporter.
I agree. I'm a critic. That's the whole point. Some of us had to argue for years and years to get this unintended consequences. The way they transform these things, rather using the gene gun or agrobacteria tumefaciens, you're randomly throwing genetic material in. You have no control over where you're inserting it. That's what insertional mutagenesis was. That's why FDA finally said in 2001, because of that, they want data on each separate transformation event. And if you go and look at the Codex Plant Guidelines, you'll see there's 4 paragraphs on unintended consequences. It took us 4 years of vigorous debate, believe me, arguing for years over wording in those areas. But yes, that is actually a very key thing. It's not just the safety of what you've put in and what it expresses. It's what else is it doing. That's the unintended consequences. That's insertional mutagenesis, and that's why we need complete molecular characterizations.
And so can I respond to that? This is 10-year-old technology he's talking about. That's not the way things are done now. When a plant is developed now, whether it's by a company or whether it's by me, we know exactly where that inserted gene has gone, because we can sequence the entire genome of a plant in a day, and that's what we do. And so as far as unintended consequences and things like that, number one, you know exactly what gene you've put in, you know exactly how many copies you've put in, you know exactly where you've put it in. And then you do RNA-seq studies, you do studies to ask about up and down regulation of other genes in the genome. So the way that, whether it's Monsanto or whether it's me, we're all doing 2015 science now. So we're doing these things in simply a much more controlled way than we did it when I started out 25 years ago. It's a completely different world. So we do have all of those details about insertion site, et cetera, and consequences to the rest of the genome.
But not for the crops on the market. And I will point out, Monsanto does not allow anyone in the academic community to do that sequence analysis to look indeed to see where those things are. Nobody. That's one of the things that they do not allow to be done. You can look at the agreements between universities and that. Yes, universities can do agronomic research, but in terms of characterization, they're forbidden from doing that. And I think it's great. All the research that is now going on, make all that stuff public, put it into the public literature. It is not there for the crops on the market. And if you look, Monsanto will not allow anybody to sequence those crops to determine exactly where it is and to see whether it's moving. You can look at the border sequences to see whether the genes are moving. That's one thing Monsanto has functionally forbidden folks from doing. I've talked to many scientists who would love to do that. They can't get approval for it.
Okay, we have we're running short on time, I'm sorry, but we have a question here on the food crisis of the world, and I'm going to address it to Xenia. GMOs are able to offer longer shelf lives and longevity in general. Being such a wasteful country, after 20 years of more studies, would it then be okay to produce such organisms, both in the US and abroad, to help with the food crisis? So I think this question is addressing whether GMOs are the key to providing food to a burgeoning world population.
So how many of you know how many of us are on this planet? A lot, right? 7.3 plus 7.5 billion people are on the planet. It is projected that that will go up to 9, 10, maybe 11 billion. But that's not really the point so much as how do we feed a world, and do GMOs play any role in it? Those of you who have a staunch anti-GMO position, prevent the exploration of GMOs being used in lots of different ways. I think many feel that GMOs are one of the tools that we want in the toolbox as we move forward. There are safety regulations, there are things, and I think the safety status is changing, and I think the amount of transparency is moving, but not everything is developed here in the United States either. There are people in South Africa that have developed corn crops using genetically modification. That was publicly funded research, and part of their concern had to do with their local corn varieties. So we need to keep this open, and I do think that as scientists, we want our toolbox as full as possible. Otherwise, we will use more land, we will destroy more forests, we will destroy more rivers and lose our soil. GMOs present one possible tool, and different technologies within that GMO category present different tools that can allow us to navigate that food crisis.
Thank you. Sheldon, do you want to respond about the food crisis?
From every conceivable critical study I've read, technology plays a small part in terms of feeding the hungry people in the world. We throw away so much food in the United States. We discard excess food. There's so many ways that we could focus the food to feeding people. We had some growth in technology and, with the Green Revolution, and that certainly helped India to some degree. But today, much of the issues have to do with getting the food to the right places, with providing land for people to farm themselves. People who don't have access to tools, don't have access to farming equipment, are the reasons that they can't even grow their own food. So we can't use technology as the magic wand that's going to solve the world food crisis.
Can I rebut?
Yes, and I just quickly say that I would point out FAO statistics show that there are twice the number of calories harvested every year as to feed everybody in the world. The reason that there's malnutrition and starvation in parts of the world is that's a political problem. That is not a problem of not enough food. We have people that are malnourished in the United States. It's not because there's not enough food. It's because of the political problems and access to land, access to for the landless laborers, access to land to grow things. So technology is never an answer. It's a political solution of dealing with poverty.
Can I rebut? So I sent a student to Malawi, and when I think we talk about food and where the food growth, the growth in population, it's Africa, it's Asia. We can feed us. We are part of the breadbasket of the world. We export food. So when I sent my student to Malawi to see what was going on, it turns out that there are technologies that can help. Not necessarily GM. Yes, there are political issues. I'm not saying that GM is the only solution. There are multiple solutions to hunger and poverty. But many of the crops that were harvested, the corn that was created, became bad because it got infected by fungi. These mycotoxins that were created made people sick. GM technologies allow us to prevent fungal disease, allow us to prevent wastage of food that is grown. And so again, for some, this is a solution. It may not be the only one. And with respect to waste here in New Jersey, I'm currently in discussions with EPA to bring a program called Food Too Good to Waste. We know about these things. We totally agree with you there that we can do better and not throw away, but do not dismiss GMOs as one tool in a multi-tool box.
We have time for only one more question from the audience. I'm going to address this to Brad. This person has concern about GM animals and fish. Can a mutation occur, and what if accidental crossbreeding occurs?
So again, not my expertise. The answer is you can always have crossing of animals, of GM animal, whether it's a GM plant or animal. You can always have genetic crossing with other plants or animals and move that gene around. I mean, that's the nature of genetics. The question of mutation is no different from the question of mutation with any other gene. Unintended consequences of an inserted gene would be no different from unintended consequences of any other gene in the genome. I guess I'll leave it there.
Does anyone want to rebut that?
Well, that's not really true. I was on the 2 expert consultations on how to do safety assessments for engineered animals, and there's actually a particular concern, particularly with xenotransplantation, and that's where they're putting genes into, for example, pigs so that they can make organs that fit into humans. And when you engineer animals, you often use certain retroviruses, and there's a concern actually that those retroviruses, or using transposons, these transposable elements, they're called jumping genes. The Mariner element, which comes from Drosophila, is one that's commonly used. There's concern that when they use them in certain backgrounds, the sort of other retroviruses that are on there can actually be activated. So when you bring in these new elements, they can actually disrupt genomes. And I should point out, through evolutionary time, humans and many organisms actually are filled with these retroviruses and other things that have jumped into our DNA, but we've shut them all down and basically turned them all off. And when you start fiddling with these engineered animals using these elements that you don't understand, you can potentially wake those things up. I would just point out that for people when you get shingles, so chickenpox is, that's a virus, and then what it does is part of it is cured, but then part of it hides from you because the virus has jumped into your genome. So it's sitting there, and then at the end of your life, if your immune system gets weakened, that virus pops back out, and that's what shingles are. So the use of the wrong vectors can actually reawaken dormant retroviruses, and that's a concern, particularly with engineered animals. So,
Okay, Brad wants 1 minute to rebut.
Just a quick thing. 2 things. Number one, transposons have pretty much nothing to do with retroviruses. They're completely different. So the Mariner elements, which I've worked with, by the way, and also with retroviruses, completely different. And having one activated has nothing to do with activating the other. Shingles, herpes virus, chickenpox, it does not integrate. So what's happening there is the shingles virus gets into your nerve cells, and then it hides there, but it does not integrate into your DNA. It is never in your DNA. What happens is you get stressed, and then the virus wakes up, just like other herpes viruses, and moves back out. But it is never part of your DNA.
Okay, thank you. Now we come to the end of our discussion, which are our 2-minute closing statements, and we're going to do this in the reverse order of our opening statements. So Xenia, you go first.
Do I get the timer?
2 minutes.
Wow, 2 minutes. So a lot has been thrown at you today. What I want you to really think about is whether you want to condemn a full technology, whether you want to close off the opportunities to exploring whether there can be health benefits, environmental benefits, using technologies that involve the genetic manipulation of crops or food or animals that may be used for food. I think that when we hear about issues with other health concerns, we need to know that that is unique to that particular technology. We can't condemn the entire technology. And so I want to encourage you to vote for GMOs, to make it a part of your toolbox. People who were at the table for organic standards said, one day, I hope we can use some GM crops because they can provide us with additional tools. I don't see them as separate. I don't see them as needing to lead separate lives. So I'm about the future, not the battles of the past, not the battles that we currently are waging, but where you want to go. And I want you to support the option of having GMOs in the toolbox based on scientific, sound studies and methodologies. Thank you.
Michael, you're next.
Okay. Actually, what might surprise people is all that I've been talking about, this is not a question of whether you're for or against genetic engineering. I'm not here saying that genetically engineered crops or engineered organisms should never be allowed on the market. All I'm saying is that there should be required, this is a new technology, there should be required safety assessments before they come on the market, and there should be labeling, and that those safety assessments should be done in an independent fashion. In terms of looking forward to the future, sure, theoretically there might be all these wonderful things that these engineered crops will do. They're not going to be part of organic because organic does not include the definition. And even if you decided to do that here, the global definition for organic, which is done through Codex, so that means if you want to ship foods internationally, that definition excludes genetic engineering, so excludes these in vitro nucleic acid technologies. If they're used, they cannot be called organic. You could call them something else. So if you want to sell organic, that's something that is not a part of genetic engineering. Sure, there might be wonderful things that GE can do. The issue is not whether we should condemn a technology and not allow it versus allow it. It's, do we allow it without required safety assessments? I still haven't heard from either of these folks from Rutgers. Do you support requiring safety assessments for these crops before they come on the market? Should FDA require safety assessments? The rest of the world, and Codex has said yes. The supporters of this technology, particularly the independent scientists, refuse to say. So I suggest you ask them, do you support mandatory safety assessments for these crops before they're permitted on the market? And then if they are, people should be able to use them freely. Thank you.
Thank you. Brad, you're next.
Thanks. Responding to that, safety assessments are done by all of the released crops to date. And all of the companies that release these crops do those, and they know that they can't release them internationally unless they do those FDA-approved safety assessments, period. GM crops aren't a single entity. Each individual needs to be evaluated individually, but the evaluation needs to come from a position of science and not of fear. American chestnuts were wiped out by a disease called chestnut blight caused by the fungus Cryphonectria parasitica. While the epidemic was raging through the Northeast, it probably started in New Jersey, just a few miles from here, before it was discovered in the Bronx Zoo in 1904. All attempts to control it failed. Some of these were quite dramatic. In fact, the U.S. Quarantine Act was a direct result of the chestnut blight epidemic. Various small breeding programs to develop resistant chestnut came and went until 30 years ago, the American Chestnut Foundation was established and introduced a back-cross breeding program crossing Chinese chestnut with American chestnut. So the result of that is the hybrids are now 15/16 American, 1/16 Chinese chestnut, and they're moderately blight resistant. In parallel, Bill Powell at SUNY in Syracuse was developing transgenic American trees for blight resistance. Of the genes he tried, the gene that worked the best and the one that he thought was would be most readily accepted was a wheat oxalate oxidase gene, a gene involved naturally in defense response in many plants. Turned out the transgenic plants expressing that single wheat gene were not only blight resistant, but Phytophthora resistant as well. So my question is, why would these trees, which are 100% American except for the single wheat gene, which occurs naturally in many plants, be seen less favorably than their Chinese American hybrid counterparts? I'll stop there.
Thank you. Next is Sheldon.
Again, this is not a debate about banning genetic engineering. It's a debate about whether or not the products are adequately regulated. The most extraordinary use of DNA sequencing is in the exculpation of 350 prisoners who were falsely accused, who spent thousands of years in prison when they never should have. So I'm definitely in favor of the use of genetic technology in many, many areas. It takes between 25 to 50 years to eliminate a dangerous chemical on the market. We have 80 to 100,000 of them. Many of them have not been adequately tested. We have to make sure that we don't go down the same road with respect to genetically modified organisms. We don't want to be where we are with chemical technology today. Glyphosate, the most widely now used herbicide in the United States, is now being more adequately tested, and we're finding that it's an endocrine disruptor. We have yet more studies to do, but what was once thought to be the safest herbicide in the world is now being questioned because the studies were not done successfully for many, many years. And I don't know what the result is going to be, but we're now learning many more health effects of glyphosate than we ever knew before. Thank you.
Okay, well, I want to thank my panelists. I hope that they have illuminated you on this subject. We could have talked for days and not just an hour and a half. There are a lot of areas that were not covered. But our question before us is, do GMOs have an adverse effect on human health and the environment? And so now it is up to you, the audience, to see whether or not you have been successfully informed. So I would like to take another vote. All of those in favor of the question that GMOs have an adverse effect on human health and the environment, please raise your hands.
I know.
All of those opposed? And all of those undecided? Aha. So there's definitely been a shift in the audience. So that, to me, means that we have succeeded in taking this from an emotional, knee-jerk reaction to a more rational debate based on more facts. So I thank you so much for your attention and for being here and for participating in this, our first Oxford-style debate. Thank you. Thank you so much.
Sure.
Thank you very much. Yeah.
You're welcome.
Appreciate all that good information.
It's great.